behavior. So that's the goal of today's lecture. So Chris, floor is yours. Thank you very much for joining us today.
Yeah, thank you, Raphael, for having me, and thanks to the organizers more broadly for giving us this opportunity to share a bit about what we do. Yeah, so today I'll be talking to you about performing coarse-grained simulations of both biological and not so biological systems. First, this kind of work is generally motivated by a desire to run simulations more quickly. Just to illustrate this point, here I'm showing a bunch of different DNA models of the same length of DNA with that are simulated for the same amount of time. And except for the all-atom, it's much smaller and actually also a shorter simulation. But the coarse-grained DNA models, you can kind of quickly see at the coarser end on the left, they relax much more quickly than at the higher resolution end on the right. And there are three reasons for this, there's three reasons for this. For how coarse-grained models speed up your simulations. One is that you have fewer interaction sites. That means generally fewer computations in order to get forces in order to update coordinates. And then when you do update those coordinates, you can typically get away with a larger time step because your particles are more massive, and also generally your potentials tend to be softer compared to atomistic potentials. And then the final speed up is that the coarse graining procedure in general results in a smoother free energy landscape. You don't have all these rough atomic details typically, and that results in faster diffusion. Otherwise, you get caught in these little local minima in the free energy landscape. So broadly, we can categorize coarse-grained models into two classes according to how they're constructed. One is top-down, the other is bottom-up. In a top-down model, you would typically take experimental data, maybe your own physical intuition to construct a model. Whereas with a bottom-up model, you might start with a fine-grained simulation, an all-atom simulation, and construct a coarse-grained model from it. Both of them will give you fast dynamics with less computation. But the top-down approach is gonna largely capture the things that you put into the model. It'll capture the behavior that you care about usually. Whereas a bottom-up model may or may not, but at least it's a systematic approach. Both of the top-down and bottom-up approaches will tend to speed up different dynamics in the system by different amounts, which means that the kinetics that you get from a coarse-grained model in general are not going to be so very reliable. And another disadvantage that, say, a top-down model might be is that your intuition or your data might have flaws. But you have kind of a similar problem with bottom-up coarse graining, where the underlying model, the all-atom model, may have deficiencies that will be kind of baked into your coarse-grained model at the end of the parameterization procedure. And then one can make the argument for either top-down or bottom-up approaches that it's a simpler process. And that which is actually simpler is going to depend a lot on the circumstances, the exact system that you're coarse-graining. Now, also, in both cases, it's not always clear what you're getting out that you don't put into the model. You kind of have to be careful not to make sure that your model can predict things that aren't necessarily just the things that you're plugging in. And then another issue is that if you want to broaden your model, add a new component or something like that, or change the salt concentration, you may be forced to reparameterize, which can be a painful process. So these are some disadvantages that are kind of general to coarse graining. Yeah. So how do you go about building a coarse grain model? That's kind of the first question. And the answer is that there are many options out there. So it's a bit like a choose your own adventure book. Hopefully it's not going to be this title. It will help, you can help avoid having the worst day of your life if you have some idea about the physical mechanisms that are present in your system. If you use existing models, that can really help, because you're not going to have to, develop your own parameters, and you can, if a referee complains about what you've done, you at least have a finger to point. Although if you do use an existing model, you should be sure to validate it. Make sure that you're applying it, that you're implementing it correctly, and that you're applying it, in a reasonable domain. If you do develop your own model, you'll want to very carefully select your target data. You don't want to overfit to one kind of data and then try to apply your model to something completely different. You'll want to carefully choose your representation. You'll want to validate, and then once you've done all these things, then you can start to make predictions. So assuming you are going to build your own model, you have to select target data. For top-down models, you're going to have to ask if you have enough data from experiment, and if it's reliable enough to use. A good example, and one that's been successfully applied in the case of the oxDNA model, which Himani will talk about especially later on, is the thermodynamics of DNA hybridization, which is shown in this kind of table, summarized in this table in the top right. For a bottom-up model, you're going to need a lot of statistics. So if you want to coarse grain something, you should have a pretty long simulation. The example I showed in the lower right here is a single-stranded DNA polymer that was simulated for, I think, a microsecond or so, which today I think would not be long enough. I would want to run longer, actually. Yeah. So, and then in both cases, you want to make sure that the conditions at which either the experimental data or the simulation data were obtained match the situation that you're trying to model. Again, you can't easily change ion concentration once you coarse grain, unless you have explicit ions in your model, or also arguably if you use a top-down model with a Debye-Hückel or linearized Poisson-Boltzmann equation for the electrostatics, then you can make changes to the concentration. But in general, changing ion concentration or solute concentration would require reparameterization. So you want to make sure that you have the right conditions in your target data. And then you don't want to necessarily fit to every bit of target data that's available. You want to make sure that you can use something to validate the model, either ideally experimental data. Okay, so then the next step, the next step is to basically select your interaction sites. In the case of a top-down model, I don't know that there's a lot of guidance for this. You kind of just pick where you want, where you think interaction sites should be. And here I'm showing the example of the oxDNA model, which reasonably uses one bead for the backbone and one bead for the stacking and base pairing interactions. Excuse me. If you do bottom-up modeling, you can be a little more systematic and select a group of atoms and map them to a site. And that has some kind of obvious advantages. In principle, you could combine the two approaches a little bit. So if you with bottom-up coarse graining, a common choice would be to use the center of mass of a group of atoms. It's not the only choice. I mean, there's other options. And I just want to comment on, and this is a little bit out of my personal area of expertise, but you can treat this process as an optimization problem, and I think there's a lot of work in the machine learning realm to try to optimally reduce the amount of information that's lost by the coarse graining procedure, making sure that you can back map and lose as little information as possible. And I think that's typically done with like a variational autoencoder approach. But yeah, like I said, it's not my personal area of expertise. So if you have interest in that kind of direction, I would check out work by Clementi and Noy and people like that. There's a lot out there. Okay, so then after you've selected your sites, it's time to parameterize. And generally, if you're doing top down or bottom up, doesn't matter. You're probably going to have to do this iteratively. Usually with a top-down model, you're not going to make perfect choices with your first attempt at parameterizing. And so you are going to want to tweak your parameters as you go until you adequately fit the target data. For bottom-up coarse graining, there are a number of different methods for obtaining parameters. I'm a big fan personally of iterative Boltzmann inversion, which is what I'll highlight here, and it's also a module of our tutorial. So the idea is basically that you run an all-atom MD simulation and you map it into a coarse grain representation. And then from that coarse grain representation, you extract target distributions. Here I'm showing the backbone, no, actually a few different angle distributions in this model. You can then take those target distributions and apply Boltzmann inversion, which is just the log of the distribution times KT, and get a potential out of it. And then generally, so you can then take your first guess at the potential based on the target distributions and run a coarse grain simulation, and you will get every time, almost guaranteed, a different distribution than the target distribution. And that's okay, because there's information in the distribution, the coarse grain distribution that you do get, and you can apply this equation in the lower left corner to then update your potential for each degree of freedom that you're interested in by how that degree of freedom is distributed in your coarse grain model. So here, rho CG is the distribution you get from the coarse grain simulation, and rho AA is the all-atom target distribution. So it basically, if your coarse grain distribution is too high in a place, well, it's too attractive and you should raise your potential there. And if it's too low in a place, then you need to decrease the energy in that region, and eventually, after repeating this process, usually around, in my experience, 25 iterations, your data should converge. It's not a guarantee, but it should. Your potential should converge to give you the distributions that you're after. So then, once you've done this, you want to validate your results. In the case of this coarse grain DNA model that I've been highlighting, we found that our radius of gyration did not match experiment after this process. We were about 10 or 15% too low. Our DNA was too compact. So that was due to the fact that our all-atom MD model was also too compact. This is this problem of inheriting the deficiencies of the underlying model. So we dealt with this by basically just adding a tiny extra bit of repulsion to our DNA beads. It was a manual process. And then after doing that for the 60 nucleotide DNA fragment, we got results that were close to the experimental values. And these experiments were obtained from small angle X-ray scattering data. these experimental estimates of the radius of gyration. Yeah, so after applying that process, we checked it for a bunch of different length of polymers, and they all were in fair agreement with the experimental data. So that was reassuring. And then we continued to validate. We applied force and extended the DNA, and we measured the extension, which is something that you can also do in experiment. The Bustamante lab had data on the force extension curve obtained for lambda phage DNA, which is a long piece of DNA, much longer than what I have shown here. And we find that we got really quite nice agreement in the high force regime, which is great. That just emerged from the model, from matching the all-atom MD simulations of this polymer fluctuating. The low force regime, we get a bit of a difference, but that can be attributed to the fact that lambda phage's random sequence in our DNA model is poly-DT. So we don't have secondary structures forming. There's no incidental base pairing happening in our model. So a bit of discrepancy at low force should be expected. And then to look at a more similar construct, we had experimental collaborators in the Ha group who looked at the fluorescence energy transfer between dyes on either end of a short single-stranded DT14 construct. And we were able to, so the FRED efficiency, this fluorescence energy transfer efficiency, depends on the distance between dyes, and we were able to successfully match their data just by looking at the distances in our simulations, except for the high salt condition at very low force. Okay, so this is sort of gives you a bit of an idea about how you might approach coarse graining. I want to talk a little bit about tools. You can implement most coarse grain models in most MD codes. This DNA model that I showed was actually originally implemented in NAMD. But we had to hack NAMD a little bit to make it work, adding tabulated potentials, because the iterative Boltzmann inversion procedure inherently uses tabulated potentials. So MD codes may not contain all the features that you want in order to do coarse grain modeling. For example, tabulated potentials may or may not be present. And so as a result, it can be desirable to turn to another code. And there's a few of them out there. LAMPS, Hundi Blue come to mind. And then we've also been developing ARBD, which is the code that most of the models that I show are being simulated with. And yeah, so there's a few options out there, yeah, that's what I want to say. So I'll talk a little bit about ARBD at this point. I just want to mention that ARBD was developed by a number of people in our lab. I think I've been the largest contributor for a while, but originally it was started by Jeff Comer. Max Belkin, as a postdoc, did some work on the code, porting it to GPUs. Hanyi continued that work for a few years while he was a graduate student. And here I'm just showing a kind of high-level view of the architecture, just to give you a bit of an idea about how things are organized. But this is a bit more on the code side. I do want to mention that we are updating ARBD. We're rewriting parts of it to try to make it easier to support a diverse set of features on diverse architectures, including multi-node, multi-GPU systems. But we've got a long ways to go still. So it's a great time to get in and say, hey, can you implement this feature? Or this would be a nice feature for you to be considering as you lay the groundwork for these updates. Okay, but regardless of which tool you use, whether it be AirBD, HuMDBlue, whatever, the simulation engine is only about as useful as the tools you have available to build these kinds of models. And so we've been working on simplifying that because AirBD historically is configured through these sort of painful to write plain text configuration files. So we've been implementing a Python framework to simplify and streamline that process for the end user. It's still writing plain text configuration files for AirBD. That's another thing we'd like to update. We'd like to have more direct interfacing between our Python framework and the AirBD data structures. Anyway, we have a couple tutorials about, first of all, AirBD model, which is very general, and I'll talk about it first. And then the MRDNA, multi-resolution DNA framework, modeling framework, which I'll highlight at the end of the talk. So first, the AirBD model tutorial covers the basics of constructing these kinds of custom models. You'll see how to make models constructed out of point particles and beads, sort of like this DNA model. It will also show you how to construct models using this iterative Boltzmann inversion process and how to set up simulations with rigid bodies. So just to highlight the kinds of systems that we're targeting, from left to right are a few simulation systems of increasing size, roughly to scale, not exactly. From this piece of DNA, we go to a whole piece of replication machinery, to a membraneless organelle, to something that's present in the envelope of the nucleus, and then we'd like to also be looking at entire bacterial cells. We're not quite there yet. With regards to the components of a cell, AirBD can help us model polymers with point particles, proteins that have some globular shape, these rigid body objects that can be decorated with particles or with density grids. We don't have a great way of modeling membranes at this time. What we would do is just confine the point particles representing the membrane in a grid potential. Yeah, and small molecules would be represented by sort of free point particles. And typically in the models that we've been producing, solvents, ions, and water are represented implicitly, not explicitly. So using this bacterium as an example, you can kind of zoom in on some component of the bacterium. Here, the yellow polymers that you see are meant to represent DNA. And there's an active, in this illustration, there's an active replication process occurring. And that's known as the replication fork or replosome. So we started a lot of this work by looking at just one tiny protein in this system, the single-stranded DNA binding protein. And we constructed a coarse-grained model around it using this iterative Boltzmann inversion process, not just for the DNA, but also for the DNA fragments, for DNA fragments binding to the protein. We performed microsecond timescale simulations of this spontaneous binding process and produced these 3D potentials, grid-based potentials, using iterative Boltzmann inversion. And at the end of that, we're able to go we're able to first of all validate that we get reasonable results by comparing to experiments, looking at the elastic behavior of these SSB DNA constructs. And then we're able to go to larger systems than previously possible, looking at the entire so-called trombone loop of the replication fork, which is this loop of single-strand DNA that gets coated with these SSBs to prevent the single-stranded DNA from reannealing. Now, our single-strand DNA is that DT model, so it's not going to be forming secondary structures anyway. But this allowed us to sort of investigate how these proteins compete to bind the DNA. These proteins are known to have a K-off on the order of days when it's like one SSB and one piece of DNA. So they're very strong binding, and it's been a bit of a mystery in the field, maybe not a mystery, but a question in the field. How exactly do these proteins get out of the way of the replication machinery? And what we observed in this simulation is that one of the proteins even temporarily falls off of the DNA completely for a short period of time before rebinding. So that competition, which we get only by coarse graining, allows us to see that kind of those kinds of dynamics. The next system I want to talk about is a carboxysome, which is this is really a study led by Day5 with Josh Vermaas's group, and we have also the experimental collaborator Cheryl Kerfeld, both at Michigan State University. So the carboxysome is this membraneless organelle that's part of the sort of the dark part of photosynthesis. It's not light dependent, but this is how leaves fix carbon dioxide into sugar. So there's a lot of interest in this process because it's rate limiting, or this system, because it contains it's where the rate limiting enzyme for the Calvin cycle is concentrated. And if you can improve the process somehow, then you might be able to create better biofuels and that sort of thing. Maybe help sequester carbon better as well. So there's some interest in this system for those reasons. So the enzyme that is the rate limiting enzyme Rubisco normally binds and reacts and causes a reaction involving carbon dioxide, but it can also instead bind and react with oxygen. So this was sort of the central line of investigation that Day5 pursued with both all-atom and coarse grain modeling. And I'm not really going to talk about the all-atom modeling that he did, but just briefly about the coarse grain models, which were performed with ARBD. So Day5 started by packing a bunch of these proteins, which have interaction potentials derived directly from the all-atom the all-atom force fields, into a spherical shell with the approximate density that previous experimentalists had noted existed. And then once he had this system assembled, he flooded it with a carbon dioxide model, also with parameters derived from the all-atom MD force fields. So it's these little white spheres that you're seeing diffusing around. And he just looked to see how often they encountered the active site of the enzymes and how often they hit the wall confining them. And it turned out that the ratio of those two things ended up being around 2.5. So for every carbon dioxide molecule that tried to exit, 2.5 first tried to strike their target site. And the reality is that so we had a confinement potential in our simulation system. The real system has almost like a virus capsid-like coating of proteins, and most carbon dioxide molecules that encounter that protein coat or shell could be expected to actually bounce off of it and have the some chance of finding a target site of one of these enzymes again. Yeah, so this slide actually summarizes a bit of the all atom results as well, which looked at the permeability of individual reactants through the shell of the carboxysome. Okay, so then the last model I want to discuss involving that's sort of highlighted in the ARBD model tutorial is the nuclear pore complex system. So this is a huge multi-protein assembly that is inserted in the envelope of the nucleus, and it is basically the gateway for cargo traveling into or out of the nucleus. Even viruses, depending on the virus, pass through the nuclear pore complex. So it consists of a sort of rigid protein scaffold. And maybe rigid is the wrong word. It's somewhat flexible, but it's structured and composed of globular proteins, and this disordered IDP mesh in the central channel. And the idea is that this mesh can filter large cargoes, prevent large cargoes from passing through, and passively allow small molecules and proteins to pass. So the central question here is, what are the mechanisms that govern the transport of proteins through this mesh? And this is work that David Winograd of our lab did, and he started by taking a cryo-EM composite structure. It's not the most. When he started, it was the latest and greatest, but today there's newer, better models out there actually. But the results we get, I think, actually don't depend that much on the specifics of the scaffold. And then he added a bunch of nucleoporins or NUPs to the scaffold, representing the scaffold as sort of this rigid potential and representing each amino acid as a single site in the disordered peptide mesh. And for that, he used a model developed by Patrick Onk. So this is realization of that principle of use other people's models, which we validated by making sure that we get the right height of a brush of these polymers, and also that the radius of gyrate, or I guess it's the Stokes radius, closely related, of individual nukes matched ones reported by the original authors. And this was implemented in ARBD, and there are some differences, of course, in the code that we have and the code that the model was originally implemented in. So this kind of validation is absolutely key. And with this model, David was able to run really long timescale simulations and observe how the density of the FG NUPs depended on the physical location in the system. He was also able to look at how they form contacts with each other, and kind of look at how likely they are to be connected depending on which NUP was present in the system. But we were interested in transport, so we threw a protein modeled as a rigid body into the system and just waited and waited and waited for something to happen. So this animation is, I think, something like 2 minutes. There are many near attempts of crossing, and this is just a small excerpt of the simulations that David ran. I'm just going to skip ahead to where it crosses. Did you see that? So it's a rare event. If you make plots of the z coordinate of the protein over time, you see many attempts. You see that there's sort of a depletion in the central part of the channel, and when it does cross, it happens quickly. It doesn't get stuck in the mesh. And David repeated this for many different proteins and got similar results for all of them. But the larger the protein, the slower and less likely crossings were observed. And for the largest proteins, we observed one cross. So our graduate student Han Yi took this analysis a little bit further by developing a method whereby you could look at just the mesh without the protein and try to determine whether or not what the time would be for a particular protein to cross. So the analysis works as follows. You basically say a protein of a particular size cannot be in contact with any bead of the mesh, and then you basically construct a map of where the accessible parts of this volume to a protein of a given size. So this map would be different for every different protein that I showed earlier. And then you get a free energy just by looking at the cross-section of this at every slice of the z-axis, of the Bohr axis, and looking at how much area is available for the proteins. So you get some density of available voxels in a given slice, and then you convert that, well, you do an average over the trajectory, and then you convert that into a PMF using Boltzmann inversion. And then the question, of course, is, how well does this work? And so the first check we did was without NUPS. And if you look, the results of the PMF from the direct coarse grain simulation, actually simulating these proteins for a few different select proteins, and then actually looking at the PMF from this analysis of the voids in a mesh without the proteins, you get pretty good agreement. With the NUPS, we still get good. Oh, sorry. We still get good agreement. Oh, well, that's fine. We still get good agreement. And so then once you have this PMF, you can do Fokker-Planck analysis to extract a mean first passage time. So this is basically another word for the Smoluchowski equation. Don't worry if those are unfamiliar terms. And then you can extract scaling relations, whatever you want. The nice thing is that we get good agreement between the coarse grain simulation and this Fokker-Planck method, but the Fokker-Planck method can be performed for very large proteins, which were inaccessible to even the coarse grain simulations. So this shows you how you can use kind of coarse grain simulations along with more kind of continuum-like analysis to in some cases go to really large systems to gain insights. So what we see is that for very small mass proteins, the mean first passage time scales almost linearly, and then there's a switch towards something exponential for larger cargos. And that basically means that there's a size cutoff almost. I mean, there's a transition region, which is shown in gray here, but but large cargoes are effectively excluded from passing through the nuclear pore complex. Okay. The big takeaways from so far should be that the coarse grain models can give you access to scales that you would not otherwise be able to reach. There are many ways to construct a coarse grain model. You should have always some kind of logic for your parameterization strategy. The details will always depend on the specific system that you're looking at, what kind of physics you need to incorporate into it. You should be asking yourself, how will I use this model to learn things that I'm not plugging into it? Usually, in my experience, that means going to taking repeating units and going to larger scales. You can parameterize a short polymer and then simulate a long polymer from that. And you should be asking how your model can be validated, because validation is key. And generally, you want to validate your results against available experimental data. And then there are many tools. ARBD and ARBD model are a couple of them that are available to you if you're interested in going down the coarse grain modeling rabbit hole. So we have a tutorial we also have a tutorial about modeling DNA nanostructures. I think I don't really have time to talk too much about this tutorial at this point, so I will skip ahead. But Amani will be talking to you a little bit about DNA. I just want to quickly say that DNA origami is a method whereby you can fold DNA nanostructures, viral DNA into whatever shape you want using synthetic staple strands that are complementary to different parts, distant parts usually of the viral single strand. And that method's been successfully applied to make all kinds of things, from cute little smiley faces to membrane channels, and even useful things like force sensing devices. And our mRDNA model basically helps designers see how to construct it helps them see how their idealized designs should actually be shaped. So typically when you design an origami, you use a special piece of software called CAD Nano, and you get these very straight structures coming from it. And this multi-resolution approach starts by using a really coarse polymer model and ends with a fine-grained one, and finally it'll export a all-atom ND model that you can use in subsequent simulation. Okay, so how do I skip slides? Okay, so so I think I don't have really time to highlight our work in this area. I'll just go to my acknowledgement slide. I just want to thank the members of my lab, our collaborators in the Vermas group and Kerfeld groups, the Chemblon group, the Ha group, and I didn't have a chance to talk about our work with Ulrich Kaiser. And of course, I want to thank the NIH resource. Sorry, it says center here, it should say resource. NCSA, Access, Frontera, Oak Ridge, and our funders at the NIH and NSF. And I'd be happy to take any questions. I think there's not a lot of time, but I'd be happy to take questions. Oh. Yep. I don't know if I can't hear anyone. I don't know if.
So that the void model of translocation, if that depends on the cross-sectional area, don't you have to ensure that the beads you're using for the coarse grain are the same size as what's actually happening in all atom simulation?
Yeah, so we used a model that I think has beads that are something like 3 angstroms. I don't remember the exact number, but it's approximately right. I think, yeah, you know, it's important that that your proteins are appropriately sized. Yeah. I think the 3 angstrom value is probably reasonable. Most amino acids don't take up a whole lot more volume than that. So you might, you know, yeah, that's a parameter you could probably play with a little bit. We actually didn't have all atom data in this case to compare to. Yeah, we didn't have all atom data available. So it's something that could be revisited, I suppose, but we were happy with just getting these sort of scaling law relationships.
Any more questions?